So, in the previous lecture we are studying the disaccharides, polysaccharides. Now the next point of these biomolecules, it is a protein. Proteins are the most abundant biomolecule of the living system. Generally, when we observe the proteins are very important for the growth and maintenance of the body. Proteins behave as enzymes. These are useful for the growth or the maintenance, or we can say the physiological process of the body for maintaining the body. The word proteins, it is taken from the proteose. Proteose means the first or the primary or the compounds which have the prime importance, which are the prime importance. It means proteins have very much importance about the growth and maintenance of the body. Generally, when we observe the proteins are, these are the biopolymers of the alpha amino acids. A large number of alpha amino acids are combined with each other by peptide bond to have the formation of proteins. When the complete hydrolysis of protein takes place, we are having the formation, or we are having the yield of a large number of alpha amino acids. Firstly, before studying the proteins, we have to know about the alpha amino acids. Amino acids. Amino acids are those compounds which have, amino acids are those carboxylic acid compounds which have the presence of amino group. That may be primary, secondary, or tertiary. Generally, when we observe the structure of these amino acids, this one is the general structure of amino acids, alpha amino acid. So when we observe, we have the presence of amino group at the alpha carbon. So these are considered as alpha amino acids. Alpha amino acid. And when we observe the D or L configuration of these amino acids, as we are having the knowledge about the glyceraldehyde molecule, which one is a carbohydrate, which one is a trihydroxyaldehyde. Pentahydroxyaldehyde, like glucose, we are having the formula here glyceraldehyde. Glyceraldehyde, in the glyceraldehyde we are having the presence of OH group at right hand side. We consider it as D. When we are having the presence of hydrogen group at left hand side, it is referred as L. On this basis, amino acids are also referred as L amino acids. This alpha amino acid, it is referred as L amino acid on the basis of glyceraldehyde molecule. Here, this one is optically active. Suppose I am writing the structure of another here. These two are the non-superimposable mirror images of each other. Here, this one is L, this one is alpha D amino acid. But in this study of proteins, the complete hydrolysis of protein gives naturally the L amino acids. Naturally they have the production of L amino acids on the hydrolysis of proteins. So these acids are not for the study of us, as naturally occurring amino acids are in the form of L. So when we observe the amino acids are have the presence of NH2 or amino group at the alpha carbon is the carbon which is directly attached with the carboxyl group. The carbonyl group of a carboxyl functional group, it is having attachment of NH2 group, and the carbon which have the presence of NH2 group, it is considered as alpha carbon. As all we know about the nomenclature systems which are used for the nomenclature of carboxylic acids, formaldehydes, or the formula for the carbonyl compounds. The carbon which is next to the carbonyl group, it is referred as alpha, and we have the presence of amino group at alpha carbon. So these are considered as alpha L amino acids. L by due to presence of NH2 group at left hand side. So these are generally considered as alpha L amino acids. Generally when we observe the amino acids are important, having an important role or have an important function in the body to maintain the pH level. Most of the amino acids are useful for maintaining the pH levels. It behaves as a observe towards the amino acids. These are stored in the form of salt in the body. Why these are stored in the form of body as a salt? When suppose when we observe the nature of these compounds, amino acids. Suppose we are having the increase in the acidic character. Suppose we are having the lower balance of the pH, or when the acidity increases. As the acidity increases, we are having the increase in the hydrogen ions, increase in the hydronium ions. Concentration of hydronium ions is increased, and this increased hydronium ions have an lone pair of electron. Have a lone pair of electron on this nitrogen, on the nitrogen of amino acid, as it is converted into corresponding positive charge. When the body have the increase in the basicity increases, means we are having the increase in the hydroxyl groups, or we are having the increase in the concentration of the OH ions. As it increases, we are having a removal of hydrogen. We are having removal of hydrogen from the carboxyl group to maintain that basicity. ज्यावेळेस आपण लक्षात घेतो त्यावेळेस हे लक्षात येते की त्यांचे ऑटोमॅटिक हे प्रोसिजर चालू असतात. NH2 ग्रुप इलेक्ट्रॉन डोनर आहे, कार्बोनिल ग्रुप हायड्रोजन आयनचं डोनेशन करतो. ज्यावेळेस बेसिसिटी वाढते त्यावेळेस हायड्रोजन डोनेट केला जातो, ज्यावेळेस ॲसिडिटी वरती त्यावेळेस हायड्रोजन ॲक्सेप्ट केला जातो. दिस इज conducted by amino acids in some cases. The more increase in acidity or more increase in the basic nature, it is not controlled by the amino acids. As we are having the disturbance in the pH value disturbance, it is responsible for the denaturation of the proteins, which in simple word we can say is the change in the structure of protein takes place. So generally these behave as basic, this behaves as acidic in the body. So this, when we observe the structure of these amino acids which are having the difference in the charge, when we observe towards the NH2 group, which is a proton acceptor group, which have the bonding of a proton. When we observe the carbonyl group, which is have the removal of a proton, capacity to removal of a proton, as we are having the removal of proton and the bonding of a proton, we are having the formation of zwitterite. Zwitterites, as we study, zwitterites are those which have the presence of both charges, those which have the presence of positive as well as negative charge on it. So generally zwitterites are electrically neutral. These zwitterites are electrically neutral, and these zwitterites are prepared by the acceptance of proton, removal of proton. These zwitterites are prepared. When we observe the alpha amino acids, this one is very important point. If we are having the presence of hydrogen atom in the alpha amino acids instead of alkyl group, this one is the simplest amino acid. This one is the simplest alpha amino acid in the all amino acids, which are common amino acids. In that common amino acids, we are having the simplest amino acid. The name of this amino acid is glycine. Glycine, which is sweet in taste, which is the simplest, which is the sweet in taste, which is the simplest. We are having the hydrogen atom instead of alkyl group. Simplest amino acid, and when we observe towards the amino acids, this glycine, it is optically inactive. This glycine, it is optically inactive amino acid. Optically inactive, or this glycine molecule is have a chiral carbon atom. The alpha amino acids other than glycine all are have the presence of chiral carbon atom, and all are optically active instead of this glycine. Glycine, it is optically inactive. It is simplest. Both hydrogen atoms are same, so this one is not a symmetric carbon and an asymmetric carbon atom, so it must be optically inactive. This one is having the absence of chiral center, so it is optically inactive. When we observe, this one is the simplest amino acid. This one is sweet in taste. हे लक्षात ठेवायचं. simplest amino acid कोणता आहे? glycine. Sweet amino acid कोणता आहे? sweetest in the taste, it is alpha glycine. A chiral amino acid or chiral or optically inactive amino acid, it is the glycine, simplest. This one is the sweetest, this one is the simplest, and this one is exceptional. This one is optically inactive. This have the presence of chiral carbon atom. This have absence of and symmetric carbon atom. symmetric alternative it is not present. We are having the knowledge about these glycine acids. But when we observe the other amino acids, known amino acids are up to 20, which are in the form of L. About that 20 amino acids, this one is the simplest, non-chiral, optically inactive, sweet in taste amino acid, sweetest in taste. Other amino acids are the neutral amino acids, acidic amino acids, basic amino acids. Also, these are classified, the amino acids are classified depending upon the presence of R. What is R? Alkyl group कोणता है? R group का है त्याच्यावर त्याचं classification केलं. If we are having the more number of, suppose I am drawing here structure of aspartic acid. It means aspartic acid. Aspartic acid. Suppose here lone pair of electrons present, one hydrogen attacks on this nitrogen. As it attacks, we are having the +1 charge on nitrogen. Hydrogen it is removed from here. It is having -1 charge. Hydrogen is also removed from here. The possibility for removal of proton, it is 2. It means aspartic acid is have increase in the charge. Aspartic acid it is having increase in the minus charge by 1 unit. It means this acid, aspartic acid or the alpha amino acid, it is acidic amino acid. It is acidic amino acid. As per we are having the knowledge about the asparagine. We are having the knowledge about the asparagine. Asparagine have, suppose removal of proton is takes place -1 charge, acceptance of a proton, which is causes the formation of the corresponding +1 charge. Here also the lone pair of electron available for hydrogen, it means it have also a +1 charge. +2 -1, it means asparagine is +1 charge. So it must be a basic amino acid. And these, the simplest amino acid, which have equal number of charges, plus it is maintained by negative charge. This one is neutral basic acid. Acidic amino acid, basic amino acid, neutral amino acid. Listen carefully. Look at carefully. When we are having the more number of carboxyl group than the amino group, then that amino acids are considered as acidic amino acids. When we have the more number of amino groups than carboxyl group, then it is considered as basic amino acid. And when we have equal number of amino acid, amino group and carboxyl group, when we have equal number of amino and carboxyl group, these are considered as neutral amino acids. These are considered as neutral amino acids. When we observe towards the nomenclature of these compounds, the amino acids are named on the basis of their sources on the basis of their taste, on the basis of their natures. Generally, 20 types of amino acids are present. So, firstly we are going to study about the nomenclature of amino acids. Draw a table. We are drawing here a table of the 20 amino acids. This is the table of the nomenclature of alpha amino acids. And about 20 types of alpha amino acids are present. This is the table of amino acids, neutral amino acids, acidic amino acids, basic amino acids. When we observe towards the nomenclature systems of amino acids, presence of hydrogen in this structure H2N, CH, R, COOH. Instead of this R, when we have the presence of hydrogen, it is named as glycine, which is having 3 letter symbol as GLY, glad, 1 letter symbol as G. Like this, when we have the presence of CH3 group, instead of in the presence of R म्हणजे काय त्याच्यावर त्याचं नेमिंग like त्याचं ठरलं. CH3 group, it is known as alanine. CH3 CH2, it means isopropyl, it is named as valine. This one is the isobutene, it is considered as leucine, isoleucine, phenylalanine, tyrosine, tryptophan, serine, proline, glutamine, cysteine, methamine, threonine, asparagine, acidic amino acids, glutamic acid, aspartic acid, also we are having basic amino acids, arginine, lysine, histidine. This is the table of amino acids. I am drawing here some asterisk markings at valine, leucine, isoleucine, phenylalanine, tryptophan, methanoid, heroine, arginine, lysine, and histidine. These are 10 number of 10 asterisk marked amino acids are essential amino acids. When we observe towards them, these are not synthesized in the body. These are not synthesized in the body. These are supplied to body through our diet. We must have a supply of these types of amino acids through our diet, and other are common amino acids which are synthesized in the body. This one is essential and non-essential amino acids. Essential amino acids are those which are not prepared in the body. When we having our meat, the body, and when we eat some parts that are digested in our body, the proteins are in the digestion. These are proteins are hydrolyzed to have the formation of amino acids. These amino acids through the blood circulation are carried towards the cells. Cells can take that amino acids and having the generation of proteins. Or the proteins are having the generation of amino acids. But some amino acids are not synthesized by this cell. Cell is not having the capacity to synthesize them. So there must be the degradation of the amino acid protein takes place in the small types of amino acids. Some changes might be there conducted in the cells. In the growth and in maintenance, we are having some changes. It means essential amino acids must be supplied to body through our diet. Non-essential amino acids are prepared in the body itself. So this one is the table of the amino acids. Here and about 20 amino acids which are well-known amino acids. These are writing I am here. These glycine, alanine, valine, leucine, isoleucine. These are the neutral amino acids, and these two are acidic amino acids. These three are basic amino acids, and asterisk marked amino acids are the essential amino acids. These are essential amino acids. So these are amino acids which are essential, which are prepared in the body, not prepared in the body, which are taken from outer sources, from other sources. Some amino acids are supplied through our body by our diet. That may be veg, that may be non-veg, depending upon that. That supply of essential amino acids is through our diet to our body. Now, the important part of these amino acids, it is their activity in the body. As we know about the general tendencies of amino groups, general tendencies of carboxyl groups, amino acid, amino group, and the tendency to bound the proton, have the space to have a lone pair of electron to balance the protons, to balance the increase in the acidic character. Acid plus base generally give rise to the formation of corresponding salt and water, like this. Amino acids behaves. These are the single letter symbol used for instead of three letter symbol. Suppose I am writing that in it. This structure of alanine. Structure of alanine. Here, if we are having increase in the proton, we will have plus one charge. If we are having the increase in the negative charge, here increase the structure. We are having increase in the plus one charge, having increase in minus one charge. Net charge, overall charge is increased. It is plus one, minus one. These are balanced by each other. These are balanced by each other, like this. These are balanced by each other. This one is this beta ion. This beta ion, which is electrically neutral. Which is electrically neutral. It means that amino acids can exist in different forms. It can exist in different forms depending upon the pH of the solutions. Depending upon the pH of solution. If the pH of solution it is low, it exists in the basic form. If the pH of the solution is increases, it exists in acidic form. It is depending upon the corresponding solution, the pH of the solution. त्याचं वर्किंग तसं आहे. जर ॲसिडिटी वाढली असेल तर ते हायड्रोजन घेतात, बॉन्ड करतात, त्याच्यामुळे त्याचं बेसिक नेचर दिसतं, ते बेसिक होतात. तसंच ते ज्यावेळेस ॲसिडिटी कमी होते त्यावेळेस बेसिक नेचर वाढतं, त्यावेळेस ते ॲसिडसारखं बिहेव करतात, ते प्रोटॉन डोनेट करतात, त्याच्यावर निगेटिव्ह चार्ज येतो. ओव्हरऑल चार्जेस आर डिपेंडिंग अपॉन दी pH of the solution. Otherwise they can behave as a deuteride. These are electrically neutral. So this one will be amino acids. In the next lecture we are going to study about the other facts of proteins, which are generally denaturation of protein, classification of protein depending upon their structure. Also we have to study about the classification of proteins. They are on depending upon the structure, like primary structure, secondary structure, tertiary structure, quaternary structures. Further we have to study about the enzymes. Also we have to study about the nucleic acids, RNA, DNA,